So in this chemistry of lipids, we are going to look at the definition of the lipids, classification, and the biological importance. So what is lipids? Lipid is organic substances that is relatively insoluble in water but soluble in organic solvents like chloroform, ether, and benzene. So generally, if you look at most of the other macromolecules like carbohydrates, proteins, and nucleic acid, they are polymeric in nature. When we say they are polymeric in nature, it means that they are reduced by the condensation of so many numbers or hundred of the monomers, hundred of monomers. Like carbohydrates is made of by a simple sugars like glucose, fructose, mannose. And in the case of proteins, the building blocks are amino acids. And in the case of nucleic acids, the building blocks are nucleotides. So, but in the case of lipids, because of its complex nature, because of its complex structures, it is highly heterogeneous. It is made up of different groups. So that is why it appears to be very difficult to define lipid based on their chemical composition. Rather, the lipids are defined based on their solubility. So lipids are generally insoluble in water, but soluble in organic solvents. That is how we defined lipids. So generally, lipids are defined based on their solubility. So they are insoluble in water, but soluble in organic solvent. And these are some of the examples of organic solvents: the chloroform, ether, and benzene. So we are now going to look at the classification of lipids. Lipids are classified into one. We have simple lipids, we have complex lipids, and then we have dry lipids. So we are now starting with the simple lipid. So simple lipid are esters of fatty acid with various alcohols. So it means that when we talk about simple lipid is made of it's an ester of F. The Fa stands for fatty acids with various glycerol. So now, for example, we have neutral lipids or neutral fats or oil. And then we have waxes. So in the case of neutral lipids, the alcohol that is used is glycerol. So in neutral lipid or oil, we use glycerol as the alcohol. And in the case of waxes, we use other set of lipids, but not alcohol. Generally, we use the high molecular weights. The high molecular weight monohydric alcohol is used. But in the case of neutral fats or oil, the glycerol is used. Okay. So now let's look at the neutral lipids. So the esters of fatty acid with glycerol, look at it. This one is a fatty acid, number one, and this one is also fatty acids, and this one is also fatty acids. So that means in this case we have 3 different fatty acids, and then we have 1 alcohol. That is why it's called triacyl glycerol. Triacyl means there are 3 fatty acids and then glycerol. So what is the difference between fat and oil? We need to understand the difference between fat and oil. Fat and oil are all under neutral lipids. They are all under neutral lipids. So we have fats. And what is the difference between fats and oil? So fats is solid at room temperature. You should get this. What is room temperature? Room temperature is a temperature of 25 degrees centigrade. So generally fats is solids at room temperature, and it's usually derived from animal. Source. And one thing about these fats, it also play important role in skin and hair. it plays an essential role in maintaining skin and hair, and it's also help in as an insulating body organs against shock. This is one of the importance of these lipids, and it also help in maintaining temperature. So fats, one is a solid at room temperature, and it's derived from animal source. And why it is solid at room temperature? Because it's made of saturated fatty acids. Why fats is solid at room temperature is because it's made up of saturated fatty acids. Saturated fatty acids. And that is why it is considered as solid, because it's made up of saturated fatty acids and is derived from animal source. And then the next thing is oil. What is oil? Oil is also an example of a neutral fat, and it's made up of unsaturated fatty acids, and that is why it is liquid at room temperature, and it's derived from plant or melting of animal fats. So if you want to obtain oil, so the oil is usually obtained from plant, or if you melt the fat from animal source, then you can get oil. So this is actually some basic difference between this. So the fat and oil, fats and oil, they are examples of neutral lipids, and they are obtained by a esterification reaction of fatty acid and glycerol. Okay, and then the next thing that we are going to talk about is, and one thing that you should understand is neutral lipids. That is why it is called neutral, because it is uncharged. It doesn't have charge. It doesn't have any charge. So this is actually example. You see, in this case here, we have glycerol. This is glycerol, and this is when you have one fatty acid. You have ester bonding. So you have one fatty acid esterified to OH group of this, and that is why it's called monoacyl glycerol. And in this case, you have also another fatty acid attached to it, and because you have two fatty acids, that is why it is now diacyl glycerol. And then when you have another another fatty acid esterified, it means that you have 1, 2, 3. You have 3 fatty acids, and that is why it's called triacyl glycerol. It's called triacyl glycerol. But at this point, we obtain chiral carbon. We have chiral carbon, because if you look at this carbon that is in this red box, it has 4 different groups. It has 4 different groups attached to it. It has 4 different groups attached to it. So as a result of that, a carbon is considered as a chiral center, or is considered as a chiral carbon. It means it's a carbon that have 4 different groups attached to it. And that is the meaning of the chiral carbon. And it can be rotated around carbon to carbon bond. So the next thing after the neutral lipids is the waxes. The waxes is also esters of fatty acids, but with high molecular weight monohydric alcohols. So in this case, monohydric alcohols is used instead of the normal glycerol. That is one of the main difference between the waxes and neutral fats. It means that they have a high molecular weight monohydric alcohol. But if you look at the glycerol, it is trihydric alcohol. It is trihydric alcohol. What is the meaning of hydric? Hydric means OH group. So in the glycerol, there are 3 OH groups. And that is why it's considered as trihydric alcohol. But monohydric alcohols, it means that there is only one OH group. And that is why it's called monohydric alcohol. An example of these waxes we have lanolin, beeswax, and wale sperm cell. And these are the examples of this. And then the next thing we have complex lipids, and that is what we are going to look at. So in the case of complex lipids, remember from the simple lipids, you said we have fatty acid and glycerol. But in the case of complex lipids, there are other groups that attach to the lipid apart from fatty acid and glycerol. It means that they also have fatty acid and glycerol, but in addition, they have other additional groups. And these additional groups are called prosthetic groups. So we have sub-classification according to the type of prosthetic group. We have phospholipids, that is those lipids that have phosphate attached to them. Then we have glycolipids. Glyco means sugar. It means those sugars that have sugar, those lipids that have sugar attached to them. That is glycolipids. And then you also have lipoproteins. That is lipid with proteins. Okay? So now we are going to start with phospholipids. So phospholipids is a lipid that contain fatty acid, that is Fa. It contains fatty acids, and then alcohol, and then phosphoric acids. So that is why it is considered as a phospholipid, because it has 3 groups attached to it apart from fatty acid. So there is phosphoric acid attached to it apart from the fatty acid and glycerol. And that is why it is called phospholipids. So this is the example. So you have glycerol, then you have 2 fatty acid attached at carbon 1 and 2, and then you have phosphates attached to it. So they frequently have nitrogen containing bases. So let's look at this phospholipids. So phospholipid is also classified into glycerophospholipids and sphingolipids. So in the case of glycerophospholipid, glycerol means glycerol. It means that they have alcohol. It means that apart from the phosphate group attached to it, can also add more other group. So if you have alcohol attached to the phosphate group, then it is now called glycerophospholipids. And then if you have sphingosine replacing the glycerol, then it will now be considered as sphingolipids. So you see, in the case of glycerophospholipids, glycerol is used as the main alcohol, and in the case of sphingolipids, sphingosine is used as a main alcohol. And of course, the choline is attached at the phosphate group. So this is the difference between glycerophospholipids and sphingolipids. A phospholipid might be classified on the basis of the type of alcohol present. That is where we have glycerophospholipids and sphingolipids. Okay, now alcohol, we are going to look at the basic or the main differences between these glycerophospholipids and the sphingolipids. So the alcohol in glycerophospholipids is glycerol, and of course this alcohol, or these examples of these phospholipids, it can be, number 1, we have phosphatidylcholine, we have phosphatidylethanolamine, we have phosphatidylserine, we have phosphatidylinositol, we have plasmadogens, we have cardiolipins. These are the examples of glycerophospholipids. And of course, for the sphingolipids, we have sphingosine as the sugar, and examples of this sphingosine is sphingomyelin. It is found in the brain cells. So this is actually the structure of these classes of the phospholipids. First, we have phosphatidic acids. So it's from this phosphatidic acid that so many different types of lipids attach to. You see, what is the difference between phosphatidic acid and cephalin? Cephalin is phosphatidylethanol. Look at it, the difference between phosphatidic acid and cephalin is just the addition of ethanol. And then also for 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 phosphatidylserine, the difference between phosphatidic acid and phosphatidylserine is serine. So serine is attached, and that is why it is now differ from. So you see that most of these lipids, if you look at them, they are derived directly from phosphatidic acids. They also have cephalate. Then we have phosphatidylinositol, and then we have also plasmalogen. So you see, the only difference is just the addition of some groups to phosphatidic acids. Okay. And then the next thing is sphingophospholipids. So what happened to the sphingophospholipids? In the case of sphingophospholipids, you see we have uh, two, two fatty acid attached to it, and then you have phosphates, and then finally choline is also attached. So you see they also have phosphates, and they have fatty acids attached to it, and then there is also choline. So this is sphingophospholipids, or we can call it sphingomyelin. So now the next thing that we are going to look at is the functions of phospholipids. What are the functions of phospholipids? Number one, in association with proteins, phospholipids form the structural component of membranes and regulate membrane permeability. So it means that in the membrane of the cell, this phospholipid is attached with proteins. And as a result of this combination, it gives membrane the ability to regulate the transportation of substances in and outside of the cell, in and out of the cell. So it's actually this phospholipid and the proteins that make the membrane to select which substance will get in or will get out of the cell. And then phospholipid participate in absorption of fat from the intestine. So generally, we don't need, when we take anything that is actually in form of fat, we don't want to store in any fat the body, because we have a reservoir, we have a places where we usually store these fats. So that is why when we have fats in our intestine, then it needs to be transported to the reservoir. Like generally where we store these fats is one, we store it in the adipose tissue, we store it in the vaginal cells, that is ovary, and we also store it in testes. So this is where we store lipids. So phospholipids have absorbed this fat in the intestine so that they should be transported where they are supposed to be. And then the next thing is they are essential for the synthesis of different lipoproteins and those participate in the transport of lipids. And also accumulation of fat in the liver, that is fatty liver, can be prevented by phospholipid. Hence, they are regarded as lipotropic factors. So they are the factors that usually prevent the accumulation of fats in the liver. Those accumulation of fat in the liver can lead to disease called fatty liver. And then we have arachidonic acid and unsaturated fatty acids. It's liberated from phospholipid and it serves as a precursor for the synthesis of eicosanoids. And these eicosanoids, we have prostaglandins, we have prostacyclins, and then we have thrombosins. And what are the meaning of these prostaglandins? Prostaglandins, they are like a messengers. They are like hormones. They are like hormones that they help in the regulation of some activities of the cell. And these prostaglandins They are usually obtained from unsaturated fatty acid. So through circulation or oxygenation of these unsaturated fatty acids, you will obtain what you call eicosanoids. And these eicosanoids, they are the prostaglandins. And what these prostaglandins does, as I said, it helps in regulating some activities of the cell. And this eicosanoid is divided into two. This eicosanoid is divided into two. Number one, this is the eicosanoids, and it's obtained from unsaturated fatty acids. They are eicosanoids, and their main functions is actually they act as a bioactive lipid messengers, and they are generated by oxygenation of arachidonic acid. Oxygenation. What is oxygenation? Means addition of oxygen. So they are generated by oxygenation of these arachidonic acids. This eicosanoid is divided into two. We have prostanoids. We have prostanoids. We have prostanoids. Then, apart from the prostanoids, we also have linear eicosanoids. And what is the difference between these prostanoids and linear eicosanoids? The difference is simple. In the case of prostaglandins, they have ring structure. They have ring structure. And the examples of these prostanoids, we have prostaglandins, we have thrombosins, and we have prostanoids. So it means that these prostaglandins, prostacyclins, and thrombosins, they are under these prostanoids. So prostanoids, they have examples, and these examples of these prostanoids, we have prostaglandins, prostacyclins, and thrombosins. And then the next, and it has ring structure. The structure is, it has ring structure. And then the second one, apart from this prostanoid, as I said, is linear eicosanoids. That is number two. This is number one. So number two, we have linear eicosanoids. So these are the second types of eicosanoids. And this linear eicosanoid is linear. It means that it is not in closed chain, it's not in ring chain, or it's not in ring form. And examples of this linear eicosanoid, we have leukotrienes. The spelling of leukotrienes is not like this. Leuko is actually like from leukocyte. We have leukotrienes. Leukotrienes is an examples of linear eicosanoids. And then another examples of these leukotrienes, we have liposins. So these are the examples of linear eicosanoids. So you see, number one, prostaglandins or eicosanoid are derived from arachidonic acids, and this arachidonic acid is an unsaturated fatty acid with, or polyunsaturated fatty acid with 20 carbon. It has 20 carbon, and these leukotrienes are obtained by oxygenation of arachidonic acid. So it means oxygenation of arachidonic acid will give you eicosanoids. And these eicosanoids are of two types. We have prostanoids An example of prostanoid, we have prostaglandins, prostacyclins, and thrombosins. And then the next one is linear eicosanoids. We have leukotrienes and we have liposins. And these are the prostaglandins. And also, as I mentioned, they are very, very important in controlling some of the activities of the cell. They are considered as a messengers. They are like hormones, but they have short lives. Because their short lives, it's just within a minute. It's not like a hormone. So they have very short lives. It means that once they are synthesized, they will just be transported to the site of action. And once they did their actions, then they will be destroyed. And that is one unique thing with these eicosanoids. Okay. And then the next thing now that we are going to look at is the continuation of the functions of these phospholipids. So phospholipids participate in the reverse cholesterol transport and thus help in the removal of cholesterol from the body. So remember, we have what we call, we have one disease condition, which is called atherosclerosis. I know that you know about it, atherosclerosis. It's a disease condition that usually occur when you have uh, too much cholesterol accumulated on the artery, so it needs to be removed. So how does it remove? It is actually removed by so many factors, and one of these factors is actually phospholipid. Phospholipid help in the reversing cholesterol transported to the artery and take it back to the adipose tissue, because it don't need to be there in the artery. So it need to go back to the adipose tissue where they are usually stored. And then phospholipid act as a surfactant that is used in reducing the surface tension. And then cephalins is an example of phospholipids, as I shown, and it's an important group of phospholipids that